Hi, everybody. Thanks very much for inviting me. I really appreciate the chance to tell you about some of the things that we're working on. The idea of this talk is essentially that I'm going to sort of get into the background of the mechanisms for the reasons that we're studying two of the things that we're doing. And this is where I work here at the Martinos Center for Biomedical Imaging, which is part of Massachusetts General Hospital and Harvard Medical School, and I don't have anything to disclose. Unfortunately, I wish I did, but I don't. So this is a paper that we put out earlier in the year that if you want to learn more of the details, the sort of technical details, this is a good place to look. We have two ongoing neuroimaging studies right now with patients. One is a PET study. So that's a brain scan called positron emission tomography, PET. And that's looking at the resting state. So in other words, patients just go there and sort of lie still, and we just see what's happening at the baseline. And the other is an fMRI, functional magnetic resonance imaging study of symptom provocation. In other words, we're looking at post-exertional malaise with that study. And it's not the only thing that we're looking at with that study, but that's sort of the important motivation. So the condition is called myalgic encephalomyelitis. And this term was coined in the Lancet in the, in the 50s. And it essentially means muscle pain related to central nervous system inflammation. So myalgia is muscle pain. Encephalomyelitis means nervous system inflammation. And the fact remains that the evidence for that is not particularly good. In fact, that's not to say it's not happening, but it just hasn't been studied in the appropriate ways, part of the reason for which is that the methods are relatively new. So this study here is Nakatomi 2014, where they used a PET study and essentially got the best evidence that we have so far. So the term neuroinflammation is actually a little bit imprecise. Technically, inflammation usually means the invasion of immune cells into a tissue, and when people use it, they usually are talking about microglia activation, which I'll explain a little bit more about, and that's what we're looking at as well. Microglia activation is a proxy for neuroinflammation. So microglia are the resident immune cells in the brain, or a type of resident immune cells in the brain. And when they're doing their job, they become activated. And that's sort of what we mean by neuroinflammation. And I'll talk a little bit about that in a moment. So this is the Nakatomi study, and I'm going to break down a little bit about why they did what they did. But what I want you to do right now is to just pay a little bit of attention. This brain right here is in what's called a sagittal section. So in other words, it's facing to this side. Here's the person's eye about here. Here's their nose. This one's facing towards you, and this one's looking straight down. But if you look right here, this is what's called the brainstem, this area of the brain right here. And I'm going to focus my talk a lot on the brainstem because I think it's really important. So here's a zoomed in image of the brainstem. Again, it's sort of in the upper, it's above the back of your throat. It's sort of, if you push straight back through the roof of your mouth, it's right there. And this is essentially what it looks like. And it's really important for pain processing, neuroinflammation, and autonomic functioning as well. And as you might imagine, those are pieces of good evidence that it might be important in this condition. So here's just a breakdown of some of the small anatomy. Each one of these represents a nucleus within the brainstem. It's a very complicated area of the brain. It's really sort of densely packed with different types of cells. And these are all initials. So the periaqueductal gray right up here is really central for pain processing. It's actually the sort of a main nucleus through which pain information comes from the body. All of these different nuclei that are named up here, Edinger-Westphal nucleus, the nucleus ambiguus, area postrema, dorsal motor of vagus, those are all important in autonomic function. So in other words, when someone goes from lying down to standing up, your body has to go through a lot of immediate changes in order for you to not just pass out, right? And if someone's having trouble with that, say with postural tachycardia, for example, which is really common in this condition, there's probably at least, the signaling in the brainstem is probably not working ideally. And then the area postrema and nucleus of the solitary tract, which are down here, are really important for neuroinflammation. So in other words, communicating the inflammatory status of the body into the brain. And this is the reason that people feel sick, actually feel sick when there's something inflammatory or infectious happening in their periphery. So I want you to take a look at this image right here, and then I'm going to superimpose it with a functional magnetic resonance imaging study that came out of Australia recently from Barndon, okay? So this is these are areas of what's called T1 signal. It's sort of a technical thing, but essentially it's a type of MRI signal. This group interpreted it as increased myelination, so in other words, increased sort of white matter wrapping around the nerve cells. That's not my particular interpretation, but if you, at the very least, something abnormal is happening here, right? And it really sort of overlaps with this area of the brainstem that we're interested in. So I'm a little bit focused on this nerve, the vagus nerve, because I think it's really important for symptoms. And this is a schematic of how the vagus nerve works. So vagus shares the same Latin root as the word vagabond because it means wandering. So it goes all over the body, and it's really densely branched. So the vagus nerve innervates every major trunk organ. And importantly, it connects up here in the brainstem at what's called the nucleus of the solitary tract. That's not important, but what's important is that it's in the brainstem. It does a lot of different things. So for one thing, it detects a type of neurotransmitter called catecholamines, which includes adrenaline or epinephrine. So for example, when you say you have an adrenaline rush, it's not the case that adrenaline from, from right above your kidneys, your adrenal glands, it doesn't just flood into your brain and make you feel that way. It's actually detected by your vagus nerve, because they don't easily cross the blood-brain barrier. So this is one of the things that the vagus nerve does. And as you know, when you have an adrenaline rush, there's all kinds of autonomic changes, right? Your heartbeat and that sort of thing change. It also importantly detects local immune signaling molecules, which is something I'm going to talk a lot more about. So in other words, when there's an infection or something problematic happening in the brain, in the body, this is how the brain finds out. It also surveys the gut microbiome. So, the more we learn, the more it's obvious that the body itself is not sterile. Every time we look in a compartment in the body, we find, some organisms. But we know for a fact, uncontroversially, that the gut is really filled with microbes. And this is one of the ways that the brain sort of finds out about the inflammatory milieu, or the level to which that microbiome has turned pathogenic. And it initiates what's called the central illness response. So in other words, the subjective feeling of being sick is part of what the vagus nerve triggers. So for example, if you take a rat and inject them with E. coli bacteria, right, the rat will stop running, it will get a low-grade fever, it won't socialize, it won't eat, unless you've cut the vagus nerve. And then it acts normal. So the brain has not learned that the body is sick. This is what the vagus nerve does. So this is part of why I think it's important. If someone's sort of walking around feeling sick constantly, it's quite likely, it's a hypothesis, but it's quite likely that there's signaling happening via the vagus nerve into the central nervous system, and that's what's causing ongoing feeling of being sick. So importantly, when the vagus nerve does detect something in the periphery, in the body, it triggers what's called a mirror response on the brain side of the blood-brain barrier. So in other words, there's something being detected in the body, and this is what tells the brain, and it tells it through activating microglia. So I'm going to tell you a little bit about that. So here's another sagittal section of the brain, just looking from the side, and here's a little schematic of monocytes and macrophages. So these are just white blood cells. Macrophages are just monocytes that live inside of tissues. So all your different tissues have different types of what they call tissue-resident macrophages, and they're just immune cells. Macrophage just means big eater. So you've probably seen little videos of these cells eating up bacteria or viruses or something like that. That's sort of their job, and they're in all the different tissues of the body. And I'm going to tell you a little bit about how they communicate with the brain. So there's this blood-brain barrier which exists in the vasculature of the brain. And we don't want to exaggerate. It's not this You know, it's not a piece of Tupperware or saran wrap that just prevents everything, right? But it is an important barrier that sort of prevents certain things from getting into the brain, because the brain actually doesn't have a super great immune system. So there has to be kind of a little bit of a barrier to prevent things from getting in there. So this is how the brain finds out what's happening in the periphery. So when pathogens are detected by these monocytes or macrophages, and so here I've just shown a couple of the pathogens that are associated with this condition frequently. So like the Epstein-Barr virus, HHV6, the Lyme bacteria, Borrelia, as well as enteroviruses. So these are all pathogens that are often associated with the onset of ME/CFS. Importantly, from my mind, they're also all pathogens that really like living inside of nerve tissue, which I think is important. So they're detected by these white blood cells. The white blood cells activate, they turn on, and then they produce cytokines, which are a protein that sort of signal the immune system response. So I've represented them here with these little triangles. But because of the blood-brain barrier, they don't just easily flow into the brain like a hormone does, right? It's sort of difficult for them to get in, because cytokines are big proteins. They're sort of blocked by the blood-brain barrier. So how does it make it in? So one reason is one of the areas that I told you about earlier when I was talking about the brainstem is called the area postrema. And that's what's called a circumventricular organ. So circumventricular, kind of around the ventricles, right? And what that means is that there's little kind of holes in the blood-brain barrier here. And it makes it a lot easier for stuff to get in. So when these immune cells detect a pathogen, they release cytokines, and they literally can just cross right through the area postrema. One of the things that's really interesting is in sick people, the blood-brain barrier starts to open up a little bit. It's a lot easier for things to get in. And we think that this might be important, for example, for post-exertional malaise. Importantly, the monocytes themselves can actually cross into area postrema. Now, this is how I personally would interpret that study that I showed you from the Australian group, but we don't really know. But the vagus nerve's job is also to act like a diffuse sensory organ in order to detect what's happening in the body, right? So in other words, when these cytokines are produced, the vagus nerve actually detects it like a nerve would detect a neurotransmitter. And it causes a signal up the nerve. Now, don't forget that the vagus nerve wanders. It's all over the place, right? So if you have a little infection in your lung, it makes you feel sick. If you have a little infection in your stomach, it makes you feel sick. It's not the case that you have to have this massive systemic infection in order to actually feel sick, right? This is part of what the vagus nerve does. And then the signal is transmitted into the brain, and it causes this mirror response. So in other words, there's a microglia response on the brain side of the blood-brain barrier. So in other words, it detects local immune signaling. Importantly, the signaling doesn't have to be infectious, so also compression. So this is part of what I suspect we're seeing with some of these craniocervical instability cases. Compression will activate glia as well as just physical injury and damage. But infection is a really common way for it to happen. And it triggers a mirror response. Now remember, this is that Nakatomi study. That looks an awful lot like signaling that happened into the brainstem, and that might be causing a mirror response on the brain side of the blood-brain barrier. This is the brainstem here. This is the thalamus here, which is a really common place for the microglia to activate next. Thalamus is really important for sensory processing. So I suspect that this is part of the reason that so many patients struggle with sensory sensitivity, where light and sound are really aversive. If microglia are activated in that area of the brain, it doesn't allow it to do its normal filtering process that it should be doing with senses. So here's a schematic of a microglia. And those are the resident macrophages of the brain. In other words, when I told you that macrophages have different tissue types, tissue-resident macrophages, microglia are the tissue-resident macrophages of the brain. In other words, they're the big eaters in the brain that deal with damage or pathogens or anything like that. So again, if they're signaled by directly by herpes viruses, which again do like nerve tissue, and they do like to travel up nerves, possibly into the central nervous system, or by cytokines signaled from other glia, they withdraw their little arms and they sort of turn on in this way that's called activated. They change shape, and importantly, they produce a whole bunch of stuff when that happens. So they produce, I've represented this here with just these little lightning bolts. They produce things that turn on other nerves. So we call them neuroexcitatory modulators. And there's a ton of them. Prostaglandin, bradykinin, ATP, nitric oxide, glutamate, a whole bunch of things that turn on other nerves. And they also produce other cytokines. For our purposes, it's important that one of the things that they do is to produce this protein called the translocator protein, because that's what we can look for with our scans. If microglia have been activated before, for example, from a really bad infection or a brain injury, they enter a state called primed. And I suspect that this might be really important for this patient community, where microglia are sort of itchy trigger finger, right? They're sort of ready for the next thing to come along, and they sort of hyper respond, I suppose you could say, although from their perspective, they're just doing their job. So in other words, if the same amount of pathogen or the same amount of cytokine were to come in contact with a primed microglia, the amount of signaling would be much more profound. And again, if microglia are primed, this could be part of the reason that people can have such a profound response to what, you know, an otherwise healthy person might not experience as so profound. So they may have really bad symptoms from a normal cold or flu, or they may have really bad sensitivity. It sort of acts as an amplifier, right? So if this is sort of turning on neighboring neurons and the microglia is acting as an amplifier, you can see why signaling across a nerve might get turned on stronger than you'd expect. So here's that Nakatomi study with microglia. So we say, oh yes, great. So evidence of neuroinflammation in this condition, that's what we've been looking for. But unfortunately, this is the exact same radioligand, in other words, the same thing that they injected, being shown after traumatic brain injury. And here's it being shown after stroke. And here's it being shown in complex regional pain syndrome. And here's Huntington's, Alzheimer's, Parkinson's, autism, depression with suicidality, bipolar, schizophrenia. So in other words, it's on from almost everything. So it's not the case that these studies are what we call discriminant validity. In other words, it's not the case that we can look at this scan and say, oh, there it is, it's the ME/CFS, because this mechanism happens in almost everything that goes wrong in the brain. So it's good evidence that there's a neuroinflammatory process, but it's not enough, and we're hoping to do some more. So with these studies that we're doing, we inject this radioligand, and we use what's called arterial blood. So in other words, there's a catheter stuck way into an artery in the arm. And the idea is that we can measure the blood as it's entering the brain to make sure of the amount of this radioligand that's being taken up. This is the stuff that we're actually measuring with our scanner that binds to that protein. Patients will be compared to controls. So here's an example of a pilot scan that we did. One of the things that's cool that we can do is we have a dual PET MR. In other words, there's an MRI machine and a PET machine together. And here's the really good spatial resolution of an MRI machine and the really good biological resolution of the PET machine. And essentially we can overlay them on top. And we can actually look right in the area that we're interested in, nucleus of the solitary tract, and see, is the signal different in patients and controls, or is that region correlated differently with other areas of the brain in patients versus controls? Because it's not just the presence of this inflammation, it's where it is that's going to matter. So one of the issues is that Nakatomi study, there are other possible explanations for what's happening. They use sort of an older radioligand. It doesn't cross the blood-brain barrier super well, so okay, let's say that the problem is just the blood-brain barrier is broken down in patients, and that's the reason for this increased signal, right? That's an alternate explanation. It could be that the general metabolism of patients is slowed down, and so that radioligand was just broken down faster in controls, and that's the reason that there was more in patients. So there's a couple alternate explanations for those data that we're trying to get around. We're using an arterial line because we think that's the gold standard. It makes the study a lot more difficult and expensive, but we think it'll make the data much more interpretable. Two other possible explanations for the Nakatomi is that they used a reference region. In other words, they compared the rest of the brain to the cerebellum. But unfortunately, we don't know that the cerebellum has no problems, right? So we're not doing that, and instead, we're using this arterial line. Okay, so in the other direction, the efferent vagus nerve is important for autonomic control. And what ought to happen is there ought to be an anti-inflammatory reflex, right? So it's, in other words, when you get sick when you're 2 years old and you have an inflammatory response, it's not the case that you just produce inflammation for the rest of your life, right? There must be something that turns that off, a reflex that actually turns that off. And this is, again, part of what the vagus nerve ought to do. In the motor direction from brain to body, this circuit actually produces anti-inflammatory cytokines. And that same motor direction of vagus nerve is what ought to be controlling, for example, heart rate when you go from lying down to standing up, which doesn't seem to work well in a large number of patients. So we think that both directions of the vagus nerve might have problems. Now again, that doesn't mean that's the core of the problem, but it certainly seems central. So that heart rate control is parasympathetic autonomic control. A large number of patients with ME/CFS fail the tilt table test. In other words, just lying on their back, being tilted up passively, and they end up passing out. Passing out is called a vasovagal response, vagus nerve. And then POTS. So this sort of problem with autonomic responsivity can be measured with the ICPET exercise test, the invasive cardiopulmonary exercise test. And this is part of what we're working on as well. So I wanted to show you something about how the brainstem works that may be important. So if heart rate and autonomic responses aren't working very well, part of the problem might actually be just physically pumping cerebrospinal fluid through the brain. And this is something that we're sort of really interested in. So here's an image of what actually happens in your brain. This is a little bit enhanced, but this is what actually happens in your brain with your heartbeat. So in other words, the brain's not sitting still. It's actually actively pumping cerebrospinal fluid through your brain. And if the autonomic response, which we think is centered on vagus, isn't working right, then this may be a problem. And importantly, if this isn't working right during exercise, when you need a little bit extra juice in the brain and body, then that might be a serious problem. Okay, and so these are just some areas where the physiology of your body may cause differences across patients and controls and how we measure their brain. But let me get a little bit into this idea of the vasculature being controlled by the autonomic system. So in other words, when you are under demand, when you're exercising or under some sort of, demand from your body, the autonomic system ought to control the vasculature of your body to constrict or open up to allow more blood where it needs to go. And this is just an old study from the 1970s just showing that phenomenon, which we know pretty well. Let me just read this to you real quick. The remarkable flexibility of the circulation in adjusting blood flow through the different tissues according to the metabolic needs, while at times preserving the arterial blood pressure and hence adequate perfusion of vital structures such as brain and heart, is achieved by the autonomic control of the peripheral vascular system. So in other words, your body ought to adjust where it sends blood depending on need. That's brain, that's skeletal muscle if you go for a walk, right? And we have reasonably good evidence that's not working so well in patients with ME/CFS. So this is what we're trying to get at with the second study, and that's the study of post-exertional malaise. We start with recruitment from Donna Felsenstein, our infectious disease doctor, We're trying to have a really tight cohort, so we do actually exclude a lot of people. And if you're among the excluded, we apologize, but I hope you understand why we're so careful about this. Those same patients undergo small fiber neuropathy testing by Dr. Ann-Louise Oaklander. We've found that about 50% of fibromyalgia patients have small fiber neuropathy, and about 30 to 40% of ME/CFS patients have small fiber neuropathy. So we suspect that this might be important. And given that it's taken from the ankle, which is the most sort of surface area you could look, if we dug deeper, the percentage numbers would almost surely go up. We just try to make it less invasive. Those small fibers are really important for autonomic functioning. And they're part of this is part of the system that causes the vasculature to respond to challenge, including when you exercise or walk around. And then the IC PET itself is conducted by David Systrom. That's an invasive cardiopulmonary exercise test, which we know is going to make patients feel sick the next day, and they very generously come back for a scan when they feel extra sick. So this is the IC PET. So essentially, they pedal on a bike at about 60 hertz, a sort of steady pace, and gradually the resistance increases on the bike. So the bike wins. They don't pedal to exhaustion, they pedal till they can't keep pace. But it's a good, it's sort of in the scheme of things, it's a moderate exercise challenge, but for ME/CFS patients, it's a lot, and it sort of makes them feel sick the next day usually. And so we sort of take advantage of that in the best way of that term, I hope, where we bring them in for a baseline scan to see what normal symptoms look like. They undergo the IC PET, and then during their PEM crash, we bring them in again to examine the effects of that PEM crash. How am I doing on time, by the way? Am I doing okay? Anyways, just let me know when I'm starting to get close. So here's that post-exertional malaise study. We do an extensive phone screen with multiple criteria. We do a baseline scan, and then in the scanner, we do mild autonomic testing. In other words, to test that autonomic system that we think is maybe at least really important in this condition. And these are really simple things like holding your breath, just for 30 seconds or as long as you can. Holding your breath increases the amount of CO2 in your blood, which ought to cause vasodilation. So if this autonomic response system is working right, that ought to look the same between patients and controls, right? And that's one of the things that we're investigating. Another thing that we do is what we call a cold vest challenge, where these participants have a vest on their body that fills with cool water. It's not frigid, it's like 50, 60 degrees, but it's enough where, again, your autonomic system ought to respond and say, okay, I'm gonna bring some of the blood from my limbs and bring it into my core to keep me warm. It's part of the autonomic response to temperature decrease. So here's our rock star physiologist and much more, Phoebe Chan, and she's doing a Doppler ultrasound of Ceci, who's one of the graduate students that we work with, to actually look at the function of her middle cerebral artery. And she does this while she's measured to all of these measures of peripheral, so in other words, the body physiology. So she's measuring the amount of gas that she exhales, she's measuring heart rate and breathing, and the idea is to see if the stuff that's happening in the periphery is also happening in the brain. It ought to be pretty well correlated in healthy people, and that's what we're measuring. So here's some actual data. This is unpublished. So what I want you to do, this is a healthy control. The color code here represents statistics, okay? So it's a correlation. So red means it's a really good correlation between what's happening in the brain and what's happening in the body, okay? And this is during resting state, where they're just lying there, right? And you can see most of the brain has a really good correlation between what's happening in the brain and the body, right? And then here's during breath hold, where the amount of CO2 has been increased. There's vasodilation because there's an autonomic response, but it's sort of equal across resting state and breath hold, right? Not a lot has changed in a healthy person. So now let me show you a couple of ME/CFS patients. So this is at resting state, right? They're not holding their breath yet. This is just walking around. They're just lying in the scanner doing nothing, right? So again, here's the healthy control with a lot of red. And you can see here's a research subject with ME/CFS where the correlation has really just dropped out. So it's not the case that what's happening in their vasculature in their brain is what's happening in the body. So there's sort of a mismatch here. Here's a patient that's, you know, pretty good, not as good as the healthy control, but still pretty good. Here's another patient where it's really dropped out. Another patient where it's, again, not as good as the healthy control, but pretty good. Here's one where it's pretty bad. And here's another one where it's sort of a mismatch. So, you know, part of the point that I'd like to tell here is the sort of heterogeneity of this condition. You know, it is diagnostically a syndrome disorder. You know, whether or not there is a single root cause is a matter of debate. I personally don't think so. And we do our best to root out alternate diagnoses. But when someone walks up to me and tells me that they have ME, I understand what their symptoms are, but I don't know what the root cause is. And I think that a big part of our task is to figure out what the root cause is for each person. Unfortunately, it makes it hard. But I actually think that that's a really important story to tell in this condition. So here's resting state, right? They're not doing anything. And here's what happens when they hold their breath. Real simple challenge. It ought to just increase vasculature in the brain and body, right? And you can see there's almost no correlation at this point. It's really dropped way off. And this patient that looked pretty good at rest now looks a lot worse. This patient, almost nothing. And then you can see here that the correlation has really dropped off. So in other words, what's happening in the brain vasculature is not what's happening in the peripheral vasculature, which tells us that there may be a little bit of a problem. Okay, and just for some more unpublished data, these are called Vircho-Robin spaces. So, you know, I don't want to make everyone panic. These are normal things that happen with aging. You can see them in a perfectly healthy brain, especially with aging. But in the ME/CFS patients, you kind of see more than you should earlier age than you should. So it's a little bit of a marker for an inflammatory process happening in the brain. Again, that doesn't mean that inflammation is the enemy. From my mind, that means the body is trying to do something. That inflammation is trying to deal with something. But they're just these little spaces between the blood vessels and the brain tissue. And they're a little bit larger in patients versus controls. Here's a couple from those same brains that we looked at before, Vircho-Robin spaces. And here's a couple more. So we're just looking, and to my mind, this is really interesting because we see them in basal ganglia, which is a really important area for movement and things like that. So we kind of see them in 3 different areas especially, red nucleus, basal ganglia, and parietal cortex. And just here's a couple more. They're just sort of more common than they ought to be, right? And it's not the case that you never see them in healthy people, you do, but we're just kind of seeing them a little bit more often. Okay. So with that, I think I would just like to thank all my collaborators. So I have 2 collaborators here, Julia and Amy, Julia Felicioni and Amy Proal. Julia's a graduate student who helps us a lot with our neuroimaging. She's a real whiz with the technology. And Amy Proal is a microbiologist who we're working with to develop a pipeline to better characterize the organisms that are in the blood and cerebral spinal fluid of patients versus controls. So I want to thank you guys especially and the rest of my collaborators. Thanks.